All right, continuing our conversation with cellular respiration. Um, so, glycolysis has just occurred in the cytoplasm. We made two pyruvates, okay? When oxygen's present, those pyruvates are going to enter the mitochondria. Um, and before the Krebs cycle can occur, that pyruvate is going to get oxidized in the mitochondria. So here you can see we have pyruvate on the left, and it enters the mitochondria. It gets oxidized to make NADH. NADH ends up being a very, very important molecule, as you'll see later on, because it's holding on to these high energy electrons, which were made way back in photosynthesis before all of this even happened. But anyway, when we oxidize the pyruvate, we make carbon dioxide, and then we end up combining it with something called coenzyme A to make acetyl-CoA. And acetyl-CoA is going to actually enter the Krebs cycle, and it's going to take part, or it's going to be kind of what gets it all, the Krebs cycle or citric acid cycle all started off here. This is just kind of a broad overview. Basically, it's telling us that when, for each, pyruvate that or acetyl-CoA that enters the Krebs cycle, we're making 1 ATP, 3 NADHs, and 1 FADH. Here's another way to kind of look at it pictorially. So we take with the pyruvate, it gets oxidized, we make it carbon dioxide, NADH, then acetyl-CoA goes into the Krebs cycle, and then it gets oxidized like at least 4 times. We make 2 more carbon dioxides, and we make a substrate level ATP. So I'm just gonna I'll go through this process, just kind of go over some major highlights, and I'm really just kind of interested that the major highlights, the major highlights more than anything else. And keep in mind too, each process each step in this is catalyzed through an enzyme. That's why we study those enzymes. All right. So acetyl-CoA it'll combine with oxaloacetate and make citrate, and then we have some exchange here, but then ultimately we get an oxidation of isocitrate. And keep in mind that's what's combined with our original products of the acetyl-CoA that came in here. But it oxidize it, so we're making an NADH. Remember, it's holding on to those high energy electrons, and then we give off carbon dioxide. All right, then we do another oxidation. We get another carbon dioxide, and at this point, that one pyruvate that started off, that was a 3-carbon sugar. It's given all of its carbon dioxides off. It has no more carbon dioxides to give. It gave off 1 with the oxidation of pyruvate, and then 2 at like steps 3 and 4. Okay, so as we're moving along, we make an ATP, and then we do another oxidation, and we make FADH. FADH is very similar to NADH. It's just slightly different, but it has a similar function of holding onto electrons. And then we do one more oxidation, and then ultimately it comes around full circle. So it started out as oxaloacetate, became citrate, isocitrate, alpha-ketoglutarate, succinyl coenzyme A, succinate, fumarate, malate, and then we come all the way back to oxaloacetate. All right, so in the Krebs cycle, we made 1, 2, 3 NADHs, 1 FADH, and 1 ATP.